Looking at day 5 here, we've got our neuromuscular system. So we are going to see, look at this, we got a huge class today. This is great. We're going to see how our nervous system is interacting with our muscular system, right? We want both these systems to kind of come together, right? They're totally separate from one another. They are not anatomically similar in any way, but they do work very intimately together, and that's what we want to have a pretty good understanding of. So objectives today, we want to understand the role of the central nervous system and how it plays like a role in human movement, the role of the peripheral nervous system and how it plays a role in human movement. And that's, we'll look at the distinctions between those two. We'll get into the specifics of the neuromuscular junction. That's where your nervous system is actually interacting with your muscular system, neuromuscular. And we'll look at the organization of different skeletal muscle fiber types. So we will get into how muscle looks a little bit different based on like how it's shaped, and the difference between like the muscles in your core that are, for instance, like keeping you upright all day, or the muscle in your heart, which is like contracting 365 days a year, versus like the muscle in like your biceps or your triceps or your glutes or your quads or whatever, right? Like your skeletal muscle, because there is sort of a difference in how those things look, on a large level, so we'll sort of look at the difference between those flavors, if you will. Then we want to talk about like how a muscle contraction works. That's probably going to be the toughest part of today's lesson. Again, this is an example of all kinds of little itty bitty microscopic proteins, and a whole bunch of like vocabulary that goes with it. This is something that we try to cover every single module because it can be a little bit technical. So that is the goal today. That's where we are. It's where we're headed. So looking at the neurology of movement, right? We do know that like your muscles are going to contract because they are under the control of your central nervous system, right? When I say control, what I mean is that like your central nervous system is making the decisions necessary to consciously or voluntarily move your skeletal muscle. Like right now, I'm using the muscles in my jaw to like open my mouth and my vocal cords, and like and like I'm moving my arms around, all kinds of stuff. Right? This is all voluntary conscious control. Right now, there is some also like involuntary control there as well, right? Like every now and then, I'm sure you guys have, you'll get a muscle that starts twitching randomly, or like have you ever touched something really hot and you try to maybe like leave your hand there, but like eventually it's almost like your nervous system's taking over for you. There's not enough willpower in the world to sort of like prevent that. Or even just like if you go to the doctor's office and they hit you with a hammer on the knee, right, and your leg kicks forward, that is still under the control of your nervous system, and it's still your central nervous system that is making those decisions. But it needs to be able to have a connection there, and that's where like the second division of our nervous system, we call that our peripheral nervous system, is interacting with that. So we're going to look at the differences between those here in a little bit. But your muscles are going to contract when a nerve impulse initiates what is called the excitation contraction coupling, which is a big mouthful of a definition. But what excitation, let's break this whole word down here, right? Excitation contraction coupling, right? If you think about like excite, right? What does the word excite mean? It means, like, it's like, I get excited, like, it's like you get amped up, right? It's, if you were to think about it, it's like, sometimes you get amped up if you turn on like a light switch, right? Like you turn the light switch on, light bursts into the room, right? You've excited that light bulb, so in us, like when we get excited, right, what that is that's causing like some type of contraction. Our nervous system is lighting something up. Okay, so if there's excitation in the neuron and that interacts with a muscle, what will happen is the muscle will then contract. Okay, and so like this excitation of a neuron creates a contraction in a muscle fiber. It's a muscle cell. And then we just call it a coupling because, you know, they're two different tissues that are sort of linked together. So that's really how your nervous system interacts with your muscular system, right? Now, obviously, there's certain injuries out there that can sometimes prevent this connection from remaining stable, you know, like an injury in the central nervous system, like if somebody gets like severs any part of their spine, you know, that is going to, you know, sever the connection of their central nervous system, you know, so like they'll probably be paralyzed in some part of the body, right? And interestingly enough, like sometimes the body can come back from that. It can, you know, create new what we call neural pathways. You know, you think about it, it's almost like when you're driving down the street and there's construction, you're like, oh, that's not gonna work anymore, and you go up, you know, you take a right and you hopefully go around, right? Your nervous system can kind of do that. It can kind of figure out a different route, which is why like some people who are temporarily paralyzed come back from it, you know. But sometimes if the connection damage is really severe, it's pretty rare to come back from something like that. And so if you think about like how your central nervous system works, this is going to be your brain and your spinal cord, right? So your central system is this section right here, the brain and the spinal cord. So if you sever anything, you know, leading up this way, if you break your spine, you know, throughout like this entire section there, what that's going to do is that's going to sever the connection for anything that heads basically down. You can think of your central nervous, actually, that's a really good picture right there. You can think of your central nervous system here. You can see like these actually control different parts of your body and do different things. So for instance, like you've got your cervical vertebrae that's in your neck. If you were to sever the connection between there, pretty much everything down here would be shut off, because you can't really, you know, it's all going to travel, signals are going to travel down. Thoracic spine is the section in here. Lumbar spine is here, and then sacrum and your coccyx is down here. This is a interesting version of this picture. The choices that they are making are very interesting. So that's your central system, and they all kind of control like different parts of your body, right? Like, so, you know, if you sever the connection up here, for instance, like in your neck, you could end up probably being what we call a quadriplegic, where you're paralyzed in both arms and both legs. Whereas like if you're a paraplegic, that's usually like paralysis in just the legs alone, and that usually happens if it's something down here. Certain parts of the digestive system are going to be non-functioning at that point, which is where like a lot of times doctors will need to, you know, oftentimes hook people up to a machine to have that happen. So it's kind of interesting, you know, what can happen with paralysis. But that just goes to show you that you want to think of your nervous system as like a series of wires, you know, and if you clip any of those wires, they're just not going to function anymore. So that's your central system. Now, we need to have a connection from this central system, you know, like literally just the brain and the spinal cord here. We need to be able to get a connection to our fingertips. We need to get a connection to our toes. We need to get a connection to our hamstrings, our glutes, our quads, like our whole body, right? And that's where your peripheral nervous system really comes in, right? Your peripheral nervous system, well, okay, see this picture here? The red pink stuff is the central system. All this blue stuff, all the stuff branching out, right, from there, that's your peripheral nervous system. You can actually see this. They've done a pretty good job of like laying this out in the bodies exhibit. Peripheral nervous system, bodies exhibit. Again, if you guys ever get a chance to go to the bodies exhibit, it is absolutely amazing. It is a little, you know, obviously it's going to be looking at anatomic stuff, and it is a little creepy and a little weird, but it is absolutely incredible. But you can see they've actually laid out the nervous system here, right? You can see there's the central system, and they're in the center, and then there's the peripheral nervous system and how it all branches out. So when you hear somebody who has like sciatica, if anybody knows anybody who gets like that, they get pain at the top of their butt, and sometimes it shoots down their leg, is what they'll often describe it as. Alexis, yeah, that's right. You get a little bit of that yourself. So what that is there's probably, and we talked a little bit about how you've got that asymmetric weight shift. These two things are linked together, by the way, because if you have a tight piriformis muscle, it often causes the asymmetric shift, and it causes the shooting leg pain. You only get it on one side, right? You don't get shooting leg on both. You don't get the shooting pain on both sides.
Not at the same time, but if it flares up, sometimes it does flare up on different sides. It just depends.
Oh, really? Interesting.
Oh. Yeah.
Huh. I mean, I guess that still could happen. But I would assume with an asymmetric shift, it would be kind of
yeah. It's more on the right side usually, but every now and again I can do something, and then the left side I'll get left side sciatica. It just depends because I drive a lot, so
yeah,
the right side because that's my driving foot. So
sure. Do you drive a stick?
No, I just drive. No, but I just drive all the time though. And I can tell when my foot starts to hurt, my leg starts to hurt, then it creeps up to the top of my butt.
Absolutely. That's the reason why I drive the RAV4 that I got. I just needed something that I could sit upright. I used to have a Hyundai Elantra, and that thing was the worst car I've ever driven. It would give me back pain. I was like, I'm not supposed to have back pain. How do I have back pain? But I was driving to Malibu every day, 30 miles to and from work. And it was killing me. So I literally was like, all right, this thing is dead to me. And I put myself in way too much debt at an early age. So this is our peripheral system, all those peripheral nerves. So if we talk about sciatica, right? Your sciatic nerve is actually a bundle. You can see it kind of branches out from the lower portions of the spine there, runs through this little hole in our pelvis, right? And there is this muscle called your piriformis that is attached to your sacrum. It attaches your sacrum to your femur there, and it's in charge of doing a little bit of external femur rotation. You can swing your leg open a little bit. Your glute is supposed to do all that work, but the piriformis is there to help a little bit. It does a little bit of abduction as well. If you think about how it pulls this way, the leg would also kind of swing a little bit out, but it's not really its main job. Your piriformis is not really meant to do a lot of external rotation. It's not really meant to do a lot of abduction. But what can happen if this muscle gets really tight? And in rare circumstances, you'll even see sometimes the sciatic nerve can run through that muscle. Every now and then people will be born that way. That is a hard fix. If you've had this pain your entire life, that could be the case. But generally what happens is this neuron, this nerve bundle, I should say nerve, not neuron, but nerve, which is a bundle of neurons, runs behind the piriformis muscle. And if that piriformis muscle gets really tight, it starts pinching that neuron. And your neurons they don't do well when they get pinched. When they get pinched by stuff, they create signals. And unfortunately, that signal is either usually a burning sensation or a numbness or a tingling, or just straight up pain that runs down the leg. And this nerve branches out. I mean, you can see, let's go back to the bodies exhibit pictures here. That's the sciatic bundle right there. Right there. See, right where these 1, 2, 3 major bundle. Is there a fourth one? I think there's only 3 that come down, and then this huge bundle here, and then that branches off. That becomes the rest of your leg. That's why that pain travels down, because pain usually travels downwards because it's going away from the nervous system. That is sort of how our peripheral nervous system functions, right? Some of it, and by the way, you can get the upper body version of this. This is a pretty rare condition. I say that it's common in older folks, but sometimes you guys can get what's called thoracic outlet syndrome, and it's basically piriformis syndrome or sciatica of the upper body. So your thoracic outlet syndrome So there's a bunch of nerves that run underneath your collarbone. And if you get really bad posture that rounds your shoulders, you get a really bad forward head posture, you sit like this passively, sometimes you'll get like a numbness or a tingling that'll shoot down the arm rather than leg, because it's the same arrangement that we see in the upper body as we see in the lower body. That one terrifies people, by the way, because how many times have you heard a doctor say, if you ever get pain in your left arm, like you're having a heart attack? And so thoracic outlet syndrome will sometimes feel like that, and it'll scare the crap out of people. And that one's not because your glute's getting over tight. That's usually because your pec minor, which are these three little fibers here that live underneath your main pectoral muscle, those get really tight, and so they pinch that. My new folks, we will go over these muscles. I know I'm throwing muscle names out here really quick, by the way. This is not the class where we are memorizing muscle names. We will make these clearer throughout time. But just kind of bear with me here with some of this terminology. So that is sort of how our neurons are going to function, right? That's how we have our central system, which is all the decision making, and our peripheral system, which is in charge of connecting the central system to the outside world, right? Your central system is encased in your skull, and it's encased in your vertebrae. Like, it's very, very, very well protected, but it needs to be able to interact with the outside world. And so it's going to send signals away, and it sends those signals away through the peripheral part of your nervous system. And then it also needs to be able to gather information, right? And so the peripheral system also gathers information and sends it to the central system. So that's how our nervous system sort of functions overall, right? So when we talk about like human movement, right, it's a very complex series of steps that begins with a decision made by your central nervous system that is then carried out by the peripheral nervous system by recruiting your muscular system. Your peripheral system is the actual connection there. So we're going to look at a couple different parts of the brain here. And like I said, I don't need you guys to have these memorized, so we're going to move through this like fairly quickly. But there's a couple different like places in your brain. One of them is what's called your precentral gyrus. It's in the front part of your brain. We can actually see that. Here's your central sulcus right here, which is sort of like the center of your brain. Your precentral gyrus is going to be like right in here. Okay, it's going to kind of live, just next to the motor cortex. And that's where a lot of voluntary movement actually happens, right? So the precentral, right, is precentral means like before central, right? It's like just on the front side of it. Is responsible for where you're creating all your moves. So your movement is in the dead center of your brain for the most part. I guess maybe not dead center, slightly front center. Like that's where most of your human movement really takes place. It's very deep in the brain, which is good, because that means that like it's one of the things that we're able to most protect, but also we need to get blood flow there, right? And so like blood has to be able to penetrate through. So again, if we have problems with like blood flow, if we have like a blood clot disorder or cholesterol problems or things like that, if that gets into your brain and then blood gets blocked and it can't reach that central system, this is why stroke causes a lot of like paralysis. You usually see it in like facial paralysis or arm paralysis. Usually it's upper body. But like you'll see like that paralysis often happen. And that's happening because like the central part of the brain is not getting the blood flow. And it's an easy, it's the center of the brain, which is protected, but unfortunately the blood flow is limited there. So the architecture of your nervous system is extremely complex. And then obviously the architecture of your muscular system is also extremely complex. So like we've got to kind of understand this overall anatomy here if we're going to understand this, right? So one of the big things that we're talking about blood flow. Why do we need blood flow? Because it delivers oxygen and nutrients, right? It delivers glucose, which is a fuel source. It delivers oxygen, which is a major, major, major fuel source, right? Remember, glucose can be broken down for energy without any oxygen, but the problem is you end up getting a critical buildup of lactic acid, and it only creates a little bit of energy at a time. It only creates you only get about 2 molecules of ATP for every 1 molecule of glucose, which is not you know, I mean, that's a great return on investment if you're investing in the stock market or real estate, you know, but like it's not that great in terms of like outputting massive amounts of energy. So what happens, you know, if we're only getting 2 molecules of ATP, where do we get all of our energy from? Well, remember, after you get that lactic acid, it can go through the Krebs cycle if we add some oxygen, and it can go through the electron transport chain if we add some oxygen. And that spits out a massive 32, 38, depending on who you ask, molecules of ATP. So a lot of energy for them. So again, we need lots of blood flow to the brain because we need lots of oxygen delivered to the brain. So your mitochondria, your powerhouses, right, which is going to, you know, take that oxygen and use that energy, or I'm sorry, create that energy, is going to be distributed not only within the muscle fibers, but also within your neurons. Okay. And so when you look at like how a muscle fiber, you look at your muscle fibers, you'll see they just got little like mitochondria kind of like straddled all over. Those are actually, I'm sorry, that was a picture of a nucleus. But if you look at the ends here, you can see there's little mitochondria, little powerhouses sort of straddling the inside of the cells, right? And the same thing would be true of neurons. You know, if you look at a neuron, ooh, that's kind of cool. The radiograph, nah, you can't tell which one's which. I'm sure somebody who's had more schooling than me would know how to read that, but there you go. You can kind of see what this sort of looks like. You can see, you know, here's the neuron here, and then there's one of its little fingers there. We'll talk about that in a minute. And there's your mitochondria. So your neurons are going to be packed full of these things as well. So your nervous system and your muscular system both take a lot of energy to run, which is why our primary like role in you know, digesting and breathing is to keep our nervous and muscular systems alive. And we're going to do that by, you know, having ATP. So here's the various sections of the brain here. Like I said, not gonna spend too much time on this, but you've got your motor cortex, which is sort of this this front section here. So there's your voluntary movement is the dead center of your brain. You've also got like supplementary movement. So like that is like your stabilizing actions, your balance is kind of located right here. You've got your premotor cortex. That's where your very complicated movements are taking place, the very front of the brain there. And then your speech formation actually lives in the same place, actually, which is kind of fun. And then we'll see some other really cool stuff. You've got like, you know, sensory information back here, like your emotions are kind of like just in the backwards part of your brain. You've also got like your auditory cortex. This is where we hear stuff and we learn stuff, right? And then this is kind of fun. This is your prefrontal cortex. This is also like where your decision making and a lot of your personality is kept, is in the center of your brain, which is where like you'll sometimes hear people say, like, I mean, how many times have you heard somebody say something and you've gotten offended at first until you took a second to like think about it afterwards? You know, like I do this all the time. Like this is like when I drive. I have kind of a temper when I drive because I just get very frustrated with people. And, you know, when I make a mistake when I'm driving, you know, because everybody makes mistakes, right? That's what I say. I'm like, oh, you're only human, Brad. Like, yeah, of course, you made a mistake. You're fine. You know, but when I see somebody else make the tiny, I'm like, what an idiot. You know, I get all mad. I get all frustrated, you know. And that is the sort of the difference, right, between like fast thinking and what we call slow thinking. If you guys ever want to read an incredible book, it's dense. It's not the most fun book to read, but it is amazing information. There's a book called Thinking Fast and Slow. Thinking Fast and Thinking. Is it just Thinking Fast and Slow? Thinking Fast and Slow. And this book is really cool. It's by this author, Daniel Kahneman, who he basically breaks down how a lot of like times like you know, when you're answering questions, I mean, we've all seen it, right? If you're watching like, you know, when you're watching Family Feud clips on YouTube, I don't know if you guys do this. I love I love Steve Harvey. So like I'll watch like a lot of Family Feud clips. And you know, he'll like, you'll get people who like ask these questions and he'll be like, you know, and the people will come up with these answers and he's like, what? And that's what your front of your brain does. It just makes very quick, very simple decisions. Oftentimes they're not the best decisions. So the back of your brain is where you make a lot of your better decisions. Like this is where you have more time to think about things and sort of consider things, right? So a lot of times, like when you get offended, that's often because the front of your brain heard something and is responding to it very quickly. But if you take the second to sort of like, you know, think a little bit slower instead of thinking a little bit faster, it can oftentimes help with like your overall decision making and stuff. It's kind of fun. I love talking about it, but it's not really what we're getting into today. But if you guys ever get a chance to read that book, I do think it's a good one. It's a little bit of a boring read, but it's a good one, like to have finished once you've read it. It took me a long time to get through it, but once I got through it, I had a deep appreciation for it, enough to, you know, recommend it. So those are the different sections of the brain there. Like I said, the cerebrum is the largest part of your brain, and that's, you know, mostly this section here, and that's all about like sensory and motor skills. Your cerebellum, which is located underneath those hemispheres, contains, this is kind of crazy. So your cerebellum, which is this very like small section, sort of deep underneath all this stuff, right? Actually has half of the neurons in your entire brain, which is kind of fun. So it's very, very small, but it has a ton of different neurons there. And that helps you like process that motor information. So you'll make a decision, and then that is where you'll sort of figure out the best way to do things. You get the medulla oblongata, which is where all your autonomic stuff is happening. The medulla oblongata is going to be located back here. That's your involuntary stuff. So various parts of the brain doing different things. Now, all of the brain is going to come together into these big central highways that we call nerves. So like a nerve, you can see here, right? This would be like, if you look, this is your C5, C6, C7, C8, and there's your T1, right? So if we look at like cervical vertebrae, this is sort of what we're looking at a picture of here. We're looking at like this section of the vertebrae there. And then if we look at the nerves, you'll see how they all sort of branch out, right? So there's your, you know, C3, C4, C5, C6, C7, right? They all kind of come out in these big branches. And you'll notice a lot of them kind of come together, right? And this is one of the ways that like your body, if you do have a stroke or if you ever are paralyzed or something like that, this is one of the ways that like, oh, well, I can't take this normal, you know, imagine like it was traveling down this route right here, and then this gets severed somehow, right? Well, what it can do is maybe it can travel up here, you know, from the C6, travel down to the same spot and then get to that connection. You know, that's one of the ways that your body creates. We call that a new neural pathway. It's your body finding a new way to kind of get the job done. That's obviously going to take time and practice, because it does take a lot of effort to sort of like, you know, get these neurons fired up and you know, functioning the way that we want them to. So how does your nervous system do all this? How does it actually create all these signals? Well, that is what we are going to look at in our first video today. All right, let's look at the nervous system first. And I don't want the first one, I want this one, right? Yeah. All right, so your neurons, and this is a big important concept. Your neurons are going to create these signals in order to be able to actually, you know, interact with various tissues of the body. And what I mean when I say a signal is, I mean, they are literally going to, you know, if we look at like a neuron, let's take a look at like how a neuron would fire, right? You guys know how like in sci-fi movies and stuff, they always make it look like this, right? It looks like there's lightning in the brain, right? We are measuring electrical activity. Your nervous system communicates by creating this electrical activity, There we go, right? Looks like friggin' lightning bolts going off, right? That electrical current is what we need to understand if we're going to understand like how we create movement. So this is going to tell you how we actually generate that electricity. And
What if everything you did, and thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses, like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between "turn up the thermostat" and "oh my gosh, I'm on fire." That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology, and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around -55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter, like serotonin or a hormone, latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at -70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about -55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about +40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them, and that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to -75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around -55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That you will find out next time. Today, you learned how your body is kind of like a big bag of batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization, and a brief bout of hyperpolarization. Thanks for watching, especially to all of our Subbable subscribers who make Crash Course possible for themselves and for everyone else. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale, the script was edited by Blake T. Pestino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe. One more thing before you leave. We like Crash Course a lot, and we hope that you like Crash Course a lot, but I kind of feel like Crash Course is only useful for a certain segment of the population. Like, once you get to a certain age, then it's good, and then forever, it can be helpful to people. But younger people, not so much. And so, we are creating Crash Course Kids, hosted by Sabrina Cruz from Nerdy and Quirky. Crash Course Kids will start out focusing on 5th grade science, but we'll keep expanding to other topics as the channel grows. Sabrina will be talking about food chains and gravity and how the sun works and how plants eat and why flamingos are pink and many other topics. Oh, and another note, teachers, you can rest assured that we've got you covered. There will be info about the standards we've used to make sure that we're doing our very best to help you out. So if you are a teacher, or you know a teacher, or you know a child, or you know someone who has a child, or you've ever seen a child, you can tell them to go to youtube.com/crashcoursekids and subscribe, and you can go do that as well if you would find that kind of content useful or interesting.
Okay, there we go. So we got a lot going on with neurons, right? They're using sodium and potassium to generate electricity. So that is what we call an action potential. Okay, so we're going to get into this here in a little bit. We're going to talk about like how these motor neurons, right, are actually creating energy, right? Every neuron in your body is going to use electrolytes to generate an electric charge. So if we look at an action potential, right, what's basically happening is it kind of looks like this, right? You can see here, there's the outer space where there's a bunch of sodium. I mean, you think about like where people tell you, don't drink salt water because salt water will dehydrate you, right? What they mean is that like when you consume salt water, there's a lot of salt and there's a lot of sodium in it. And what that actually does, that actually pulls fluid out of your cells, because nature is trying to like spread out all of those salt molecules, and it can spread them out if it like pumps enough water in there. The concentration will go down. So we're just trying to like make some space there. So we always have a little bit of sodium. Yeah, question. What's up?
Okay, so you said sodium water just makes you dehydrated, right?
Yeah.
So like Gatorade stuff, like they have sodium on it. So it's like sodium and sugar, but why that doesn't, that's it. Yeah, so everyone who dehydrated, they say, oh, drink Gatorade or like that.
So here's the thing. So there is actually sodium. The active ingredient in Gatorade is sodium citrate. If we look at Gatorade, right? What this is it is 10% sodium and 1% potassium. So there is sodium in Gatorade for sure, no argument there. However, it also includes monopotassium phosphate, aka potassium, right? So it's got some potassium in there as well. And when I'm talking about these electrolytes, you see like what we're seeing right here? See the Na, and then it's got a plus sign?
Yeah.
K that also has a plus sign. So that Na molecule, that's sodium. And then this K right there, that's potassium, right? So that's the chemical symbol for potassium. So inside of our cells, we have a higher concentration of potassium. Outside of our cells, we have a higher concentration of sodium. When you drink Gatorade, the idea is that it actually helps hydrate you because it is adding both of these electrolytes. Some of the potassium can rush inside, and the sodium is not high enough that it actually pulls water out of our cells. It actually can still stay just outside there. If we look at like, let's see here, what concentration of seawater is salt? 33%. So salt water is about 33 to 35, up to about 37% sodium, right? That's way higher. So like, you know, we are mostly filled with water, and some of the water that we are filled with is pretty salty. I mean, you've probably cried at some point, you know, like it's pretty salty sometimes, right? So that is like, we do have like sodium, but if our concentration of like what we bring in is super high, even that kind is like your nature basically is going, wow, that's way saltier and that's less salty. So it just pulls the water into it naturally. Gatorade is not enough sodium to have that happen. In fact, it's actually just the right amount of sodium that actually increases hydration. And that's the theory behind like electrolyte water. Smart water has electrolytes in it. It's basically water that has like a little bit of sodium in it.
Okay.
Yeah. So when I say electrolytes, guys, you know, that's sodium and potassium. Those are the two big ones I want you to remember. So sodium is an electrolyte. Potassium is an electrolyte. And they both have, this is what is somewhat confusing. They both have a positive charge, but potassium has a way more positive charge. I'm sorry, that's not true. The opposite is true. Sorry, sodium has a way more positive charge, and potassium has less of a positive charge. So if you look at the difference between the two, if you look at the voltage, right? Sodium is very positive, and potassium would be considered negative in comparison. So what nature can do, right? Think about like the last time you took the batteries in your remote control, right? Have you ever put your batteries in both facing the same direction, right? Nothing happens. It can't create electricity. Like the way that we capture electricity to run like all of our electronics is we're basically, you know, using that battery. So you take like two batteries, you know, and you put them side by side, right? And then you would put like the positive end over here, and you put the positive end over here. And so now what you have is you have like a positive end, the positive end on this side, and you have a negative end over here. So positive and negative. And what's going to happen is this stuff is going to want to interact. This wants to go to here. This wants to go to here. So what's happening is that electrons are basically bouncing back and forth between these over and over and over and over and over. So those electrons are just passing back and forth, trying to stabilize, because again, nature wants to even things out. And so then the electronics that we're going to use, like your battery, like your TV remote or your car battery or whatever, they're basically just going to capture those electrons as they're running by. And so we're going to grab them and use them to power our devices. And that's how we run electricity on, that's how batteries work, right? This same thing, which I don't know anything about batteries, but I do understand biology. And like, this is how like our body uses that, right? Our body basically has a positive side and a negative side. And when a stimulus happens, some type of stimulus triggers the movement of these materials. If we were to push sodium from one side of the cell, if we had it rush inside of our cell, nature can't stand that. Nature does not like it when there's a lot of, you know, when all this positive stuff is like matched together. So what it will do naturally is it will push potassium outside of the cell. And now what you have is you have a negative end here, positive end here, and this is polarized. This is the opposite of what it should be, because this is still very positive and this is still very negative. So now electrons are going to go rushing back and forth like this, And our body's and that's going to create this electricity. It's going to create this lightning bolt, right? And so then like our body is going to, capture that energy, and it's actually going to travel across. So all of the pumps are going to change their shape. So let's look at sort of what this looks like here. You can see in this sort of like animation, right? The action potential is traveling from one end of the neuron down to the other end of the neuron. And then eventually it's going to synapse with a muscle fiber, right? So it's basically just switching. There's what we saw from the video, right? It's switching the positive, negative association, and it's traveling back this way. Sodium sort of rushes back this way until eventually it gets restored, and then the next signal can happen. So if we send a bunch of these signals very, very, very quickly, that creates a very strong, very powerful muscle contraction. And if we send these signals very slowly, one after the next, kind of slow, that creates a very weak muscular contraction. And that's how your body controls the strength of your contractions. So when you're going to the gym, lifting very, 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 very heavy, what you're doing is you're sending signals like this, right? Whereas like if you're lifting at like sort of an endurance weight, where you're not lifting very, you're not recruiting all of your muscle fibers, you're maybe only recruiting a few of them, it might be a signal like this. It's a little bit slower. And that's how our body regulates the strength of our contractions. We can pick up, a puppy gently, and then we can deadlift a barbell explosively, not gently, if that makes sense. And here you can even see, oh, that is just the fastest GIF ever. But you can see, right? Like, oh, not really. You really can't see. That's so freaking speedy. Give me a slower one. Friggin' things. There we go. So you can see, like, sodium is going to go rushing in and potassium is going to go rushing out. So that's how these little pumps actually make this happen. So luckily, all of our cells have these little pumps that move these concentrations back and forth in order for us to be able to create these, we call them an action potential. Okay. This is how we create that electricity. Does that make sense to everybody? Is everybody fairly clear on that? I know, like, I still have questions about how this works. Sometimes I'm like, what really causes like the initial, thing? How do we move it the first time? And I'm sure somebody knows the answer to that. But just in terms of like moving sodium and potassium, right? Those things are opposite of each other. So it creates electric charge. And as we move that electric charge, it'll go from neuron to neuron to neuron to neuron until eventually it interacts with a muscle cell. And when it gets to that muscle cell, then it'll cause electricity to run across the muscle cell. So we'll actually get an action potential in the muscle cell. And that's going to trigger muscular contraction to take place. Everybody pretty clear on that? Any questions about how that works? Anything I can do to make that clearer?
No, that's pretty easy. That's how they get remote control cars to move too.
Yeah,
it's like
100%.
Just move magnets around. That's pretty much how your muscles work.
Yeah, magnets are also very, very similar. That's the reason magnets are attracted to one another, is because the electrons are attracted to one. They're trying to move from here to there, and so then it causes them, and that's why they have a polar side that repels, because that's pushing the nature's going, no, we don't want those electrons next to each other, and then it has like the unpolar side, which is, attracting them, the attractant side. But yeah, magnets, same situation. It's all electrons moving. You can create an electromagnet actually pretty easily. You just have to take a magnet and cool it down to super freezing cold temperatures. And it'll actually have a little bit of an electric charge. If that were if that were actually an efficient way to do things, we would solve the world's energy problems. All right, so let's go ahead and talk about where this starts interacting with muscles, because we really haven't even dived into our notes yet, right? Let's get into how we actually interact with muscles. So let's take a look at like what our muscle anatomy looks like. We're going to come back to this nervous system thing in just a moment, but let's look at a muscle fiber here. When I say muscle fiber, I mean muscle cell. Okay, so I want you guys to picture, if I look at just like, go to your classic high school biology cell, right? Little, sort of a little bubble looking thing. Now, for the record, there's not a single cell in your body that looks like this. Yet this is the cell that we all studied in school to learn. This is a diagram cell that's meant to like show you everything that could be inside of a cell, but none of the specialized stuff. This is the stuff that pretty much every cell has, right? So it's got a nucleus, it's got a little mitochondria, it's got a membrane around the outside, lysosomes, which are gonna, clear out the body and clean things. It's the immune system for the cell, basically. Little vacuoles and reticulums and stuff like that. All these like different things, right? Muscle cells are gonna be special though. If we look at a muscle fiber, look at an individual muscle cell, it looks a little bit different, right? You'll notice, it's going to kind of look like this. You can see like the nucleuses are actually kind of straddled around the outside of the cell. And then what the heck are all these little rods? Like, look at that. You don't see that in a typical cell, right? I don't see any rods running through this little bubble here, right? Well, that's what makes muscle cells very unique. They're these big, long, kind of tube-looking things, and that tube is filled with these specialized little proteins, these specialized little rods, and those rods are called myofibrils, and that's where the magic of contraction takes place. These little rods that run from end to end in a muscle cell. So then you'll see all the normal stuff in there as well. You can see, like, remember I mentioned there's a reticulum, right? Well, there's the reticulum right there. It just looks a little bit different. It kind of surrounds the rods, actually. There's the mitochondria, right? Little mitochondria kind of just like, shoved in between each of the rods, right? So there's the nucleus, and there's this special stuff called T-tubules. There's all kinds of special materials. And that's another thing that's like kind of true about the universe. I always tell people, you want to understand the universe, nature hates a gradient. That's one thing to remember. Man, we just mentioned that, and I didn't get that from Crash Course. This is what everybody says, right? The other thing you want to remember about the universe, guys, is that form follows function. What I mean by that is that, like, if something is shaped a certain way, it will act a certain way. If if you've ever heard somebody say, like, fake it till you make it, it's actually really good life advice. If you shape your life to start, to be similar to that of like somebody that you admire, like a successful person, let's say there's a millionaire out there that you admire, and you're like, I want to shape my life like that. I want to do the things that they did. Guess what? You shape your life that way, it's gonna turn out that way. Like, this applies to not only like, I'm not giving life advice here, but this is how the entire universe. Bone cells stack very efficiently. That makes bone tissue very hard. Their form reflects their function. Your muscle cells are unique in their shape, in their form, and that allows them to contract. That's what gives them their special function. It's what makes muscle cells special. It's what makes neurons, their form is that they have sodium and potassium pumps. They have a ton of them. And those sodium potassium pumps are what allow electricity to be able to get generated. Form follows function. So you want to understand the universe, just remember, if you're ever unhappy with the way something turned out, just remember that it was crafted that way. And if you want to change it, then you got to change the shape. Pretty good analogy for working out, right? So inside our muscle cells, you can see all these specialized little rods. Those little rods are called myofibrils. So a myofibril is a rod that houses these little proteins that are going to do all the contractions. So you can actually see those are the proteins right there. Now this is actually pretty zoomed out, believe it or not. Like, I know we're zoomed in on it. This is an individual, like a piece of an individual cell. But look at this. You can see there's sort of this overlapping that's happening there, right? You can see it's kind of like this is overlapping with that. It sort of looks something kind of like this. There's an overlapping action happening here, right? Between like this central one. So if I were to make this just like a little bit thicker, right? We'll make this one a little bit thicker to sort of make it distinct from the other one, right? So what you're going to see here, and I'm just going to go ahead and actually like, let's put another one of these down here. Match that up. And one more in the middle. There we go. So now we can kind of see there's this overlapping action that's sort of occurring here, right? This overlapping of these proteins is what's going to be able to happen here is if I were to take this protein, right? If I were to take this protein right here, and I wanted to slide it this way, and I took this protein and I slid that one that way, right? This is tied end to end in the muscle. So when I slide those proteins closer together, it pulls the muscle cell from this side. It grabs it and it pulls the muscle cell from that side, and it pulls it towards its center, right? If these proteins crawl towards each other, they crawl towards the center there, that's going to cause the entire muscle cell to go from being this long to being this long. And that is how your muscle cells actually contract. They do it because they have these overlapping little proteins here. So that's what we're seeing in this picture. We're seeing a little overlap there. So like what's going to happen, if I go ahead and just like steal this image here, right, is that this stuff, these little red proteins, you know, here are going to move this way. And then these are going to move this way. and they're going to pull in that direction. And that's going to make this section of the myofibril shorten. If that happens in the next section of the myofibril, like over here, that's going to shorten, and that one's going to shorten. And the whole rod is going to get shorter. And as that rod gets shorter, the entire muscle cell gets shorter. And as that happens in all of the muscle cells, the entire muscle shortens. That's what you're seeing. When you look at like a muscle bunch up, right, and it creates that kind of like bunched up action, that's because all these little proteins are sliding across one another. So those proteins, let's go ahead and understand what they are. So myofibrils are the rods themselves, and myofibrils house what we call our myofilaments. Myofilaments are specialized proteins that are able to bind to one another, and they create that contraction. So you've got thin proteins. These are called your actin. Okay, so that is actin. And the thick proteins right here, that is called your myosin. Now, if you actually look at actin, it actually looks something kind of like this. This is going to take me just a second to whip up a picture. But actin strands actually look something kind of like this. We're going to zoom in just a second here. Whoops, didn't want to do that. Sorry, give me 1 second to kind of whip up this picture. Oh, what am I doing? I can just go like that. Okay. So there's our strand of actin. I need another one. We'll put that one here. We'll put another one over here. And we'll put another one over here. Okay, so now if we zoom in, that's what this thin strand, this thin actin strand, you know, that we're seeing like right here. This is what they look like. They got these little like binding sites on them, right? And then there's that really thick band in the center there of myosin, right? So I'm going to go ahead and just, you know, take this whole thing and copy that, and then make it like even thicker and bigger. So there we go. So now we've got the thick myosin strand it kind of looks something like that. But it doesn't just look like that. What it actually has is it's got these little heads on it that look something kind of like this. They kind of look like this. They got these like little like arms, and then they kind of go like that. And then they got this big bulbous head on it. All right, and just making proteins here. All right. Now I just need to be able to flip that. Oh, come on. How do I do this? Sorry, guys. 1 second here. Vertically. There we go. So it looks like that on that side. Yeah. Looks like that on that side. And why can't I figure out how to do this? You know what? I'm just going to draw new ones. We're almost done anyways. There we go. All right, so that's actually what they look like. They got these little like heads that reach up on them. And what they want to do is they want to go from this shape here. They want to reach up and grab the binding site, and then they want to straighten out like this. And what that does is that moves the actin strand that way. So we want to grab this and pull the actin strands towards the center this way. So like all of these guys on this side, all these guys on this side are going to pull towards the center that way. And all the guys on this side are going to pull towards the center that way. That sliding action that's happening in the actin and the myosin, right? You know, they're both pulling towards their center like that. And so the entire section is shortening. This area right here, by the way, if we look at just like, you know, if you notice a myofibril, you know, they are just a whole bunch of these things all tied end to end to end to end to end, right? You can kind of see, right? Like, see how like these are all here, and then like there's this section, and then there's another one over here. There's this section, there's another one over here, another one over here. There's a ton of these little sections, these little overlaps that are all like tied together end to end until you get to one side of the muscle cell and you get to the other side of the muscle cell. So like a myofibril is just nothing more than a whole bunch of those, you know, things crossing over another. This area, by the way, like from here to here, we call this the sarcomere. So the sarcomere is where the overlapping takes place. So again, we're going to see this sliding action in these two directions here. It's going to pull towards its center. And then we would just line the next one. We would just line the next sarcomere up directly side by side, this other one, right? Like I would just go like this and go like this. And I would just put another one like right by. The only difference is I wouldn't line it up like that. I would line it up slightly straddled. And that's going to create this sort of zigzagging action. See how it kind of creates like a Z action there? That area there is called a Z line. And Z lines are sort of indicating where you have, you know, separate sarcomeres there, right? So the sarcomere is the area between two Z lines where they both contract towards their center like that. So Let's go ahead, and I know like this has been a lot of anatomy and stuff, but this is where we got to watch another video that's going to actually show you all of these proteins and watch this sliding action. But this is what we're looking for. We're looking at the sliding filament. You know, if we're looking at how these proteins are binding up, this is what it looks like in all the sarcomeres simultaneously. Every one of those myofibrils slides the little actin strands towards their center. They kind of reach up and they kind of grab. It's not a GIF. They reach up and they grab those little proteins. Sliding, you can see the purple is sort of sliding further in, right? So it grabs them and pulls them towards the center. So how the heck does that happen? Well, it happens because your nervous system tells it to happen. When electricity runs through a neuron, it triggers like electricity to run down that neuron. When electricity runs through a muscle cell, it triggers contraction. That's the difference between neurons and muscle cells. So like I said earlier today, right, these things have a lot in common. They're very similar to one another, but the anatomy is very, very, very different because that's going to cause their shape is going to do different things. So here's a little video kind of highlighting this. Crash course muscles. And that is not the one I want. I want this one.
Stop overpaying for sunglasses. It's crazy that the most expensive thing you're wearing.
Romeo and Juliet, Helen and Paris, Tristan and Isolde. These famous star-crossed lovers bring to mind insatiable longing, forbidden love, and tragic separation, and poets and emo rockers love them for it. But you know where else you can find a nice, hot romance? Your muscle cells. They've got their own famous coupling, a pretty pair of tiny protein strands called actin and myosin. Romeo and Juliet may have set a chain of tragic events into motion with their infatuation, but deep down in the cells of your muscles, the hot protein action between actin and myosin is actually literally causing all of your motions. All of them. And I don't just mean voluntary stuff like walking down the street or moving your mouth so you can talk or chew chips, because your muscles also support your weight and help fend off gravity. The amazing thing about your complicated, self-healing, blood-guzzling muscle tissues is that they turn chemical potential energy into mechanical energy, or movement, simply by doing two things: contracting and relaxing. And that contracting and relaxing is exactly what's fueled by the constant coupling and separation of biology's greatest lovers. Somebody get these proteins a movie contract. You will recall from our early lessons on tissues that you're kept alive and moving by three types of muscle tissue: smooth, cardiac, and skeletal. Your smooth muscle tissue is found in the walls of all your hollow visceral organs like your stomach and airways and blood vessels, where it involuntarily and very usefully pushes fluid and other material around by contracting and relaxing over and over. Your heart is so important that it gets its very own muscle tissue type, cardiac muscle, which looks striped or striated and also functions involuntarily to keep your blood pumping without you having to think about it. But when you hear the word muscle, you're probably thinking of the kind that you see on Chris Evans when he first walks out of that machine in Captain America. And those types, the ones you can see and feel and flex, are your 640 skeletal muscles. They're striated like cardiac muscle tissue, but they're also mostly voluntary, meaning you have to think about using them and activate them with your somatic nervous system. Most of them attach to your skeleton and create movement by pulling bones in different directions as they contract. Each one of your different skeletal muscles, like your biceps brachii or vastus lateralis or gluteus maximus, is technically its own organ made up mostly of muscle tissue, but also of connective tissue, blood vessels, and nerve fibers. And because your muscles are voracious energy hogs, each one is rigged up with its own personal nerve to stimulate contraction, and its own artery and vein to keep it well fed with all the blood and oxygen and nutrients it needs to operate. But to understand those operations, we first need to get a grip on the anatomy of a skeletal muscle, which involves fibers within fibers and lots of layers. Basically, a skeletal muscle is constructed like a really sturdy sturdy piece of rope. Thousands of tiny parallel threads called myofibrils squish together to form muscle fibers, which are your actual muscle cells, cells with mitochondria, multiple nuclei, and a cellular membrane called a sarcolemma. Those muscle fibers then form larger string-like bundles called fascicles, which combine to form the larger rope-like muscle organ like your biceps brachii. Overall, this bundles of bundles configuration makes muscle tissue fairly sturdy, but considering how much abuse your muscles take when you do something like pretty simple, like lift a big bag of dog food, it's no surprise they need a little help. That's why every muscle contains a few different kinds of supportive sheaths of connective tissue, the protective reinforcements to keep that bulging muscle from bursting. So that is the structure part of the story. But if you want to get into the nitty-gritty, the down and dirty of how you actually move, well, there are rules. Really just two main rules, and they have to do with proteins. And they're both true for a lot of the proteins we talk about, whether they're enzymes or ion channels or receptors or muscle proteins. And these rules are: 1, proteins like to change shape when stuff binds to them, and 2, changing shapes can allow proteins to bind or unbind with other stuff. So keep those rules in mind while we see how a muscle fiber contracts and relaxes. Now, remember those tiny myofibrils that bundle up to form your muscle fibers? They're divided lengthwise into segments called sarcomeres, which contain 2 even tinier strands of protein, 2 different kinds of myofilaments called actin and myosin. And it's their angsty story of star-crossed love that fuels every movement your body could possibly dream up. A sarcomere contains both thin filaments, made up mostly of 2 light and twisty actin strands, and thick filaments composed of thicker, lumpy-looking myosin strands. Each sarcomere is separated by what's known as a Z-line at either end, which is just a border formed by alternating thin filaments in a kind of zigzag pattern. A muscle contracting is all about sarcomeres contracting, bringing those Z-lines closer together. Alright, so now comes the romance. When your muscle cells are at rest, your actin and myosin strands don't touch, but they really want to. Specifically, that club-headed myosin wants to get all up close and personal with the actin. When this happens, and it will eventually, it's called the sliding filament model of muscle contraction. But in the meantime, like in any good love story, the pair have some obstacles to overcome. Namely, actin is blocked by a couple of protein bodyguards called tropomyosin and troponin, which keep getting in the way. Luckily, these guards can be bought off with a little ATP and some calcium. I prefer cash and nachos, but whatever. Remember, ATP is kind of like molecular currency. It contains chemical energy, and your muscles are all about converting chemical energy to motion, so they're always hungry for more ATP. Your muscle cells have lots of nuclei, but some of them also have a lot of mitochondria, whose sole purpose in life is to crank out ATP. And muscle cells also have their own version of an endoplasmic reticulum, the cell's transport and storage system, but in this case it's specialized, so it gets a special name, the sarcoplasmic reticulum. Its walls are loaded with calcium pumps, which use ATP to save up a bunch of calcium ions. And it's also studded with calcium channels that are linked to voltage-sensitive proteins in the membrane of the muscle cell. Say I want to move my arm. My brain sends an action potential along the motor neuron until it synapses with a muscle cell in my arm. The receptors on that muscle cell are ligand-gated sodium channels. So when the motor neuron releases our old friend acetylcholine into the synapse, the channels open up and create a rush of sodium into the cell as a graded potential, which, if it's strong enough, causes nearby voltage-gated sodium channels to open. Now I want to take a second and point out here that we're still talking about an action potential, but not in a neuron. This is happening in a muscle cell, people. So that action potential zips along a muscle cell's membrane, the sarcolemma, which has lots of tubes that run deep inside the cell called T-tubules. When the action potential travels down one of those tubes, it eventually triggers the voltage-sensitive proteins that are linked to those calcium channels in the cell's sarcoplasmic reticulum. When those channels are thrown open, the calcium stored inside rushes into the rest of the cell, and finally myosin is like, yes, here we go! At this point, the myosin is totally stoked, because the bodyguards that have been frustrating it are in for a big, irresistible distraction. That's because the protein troponin just loves to bind with calcium, and remember, when stuff binds to proteins, the proteins change shape. So the calcium latches onto the troponin and causes it to pull the other bodyguard protein, the tropomyosin, away from the sites on the actin strands that the myosin really wants to get its paws on. And suddenly it's all, okay. But the only myosin heads that can bind to those newly exposed sites are the ones that are ready for action. That is, the ones that have already grabbed a molecule of ATP that's been floating around and broken it down into ADP and the leftover phosphate. When a myosin head does that, it moves into an extended position, kind of like a stretched spring, still holding on to the ADP and the phosphate, and still storing the energy that was released when they were broken apart. So after all that, with the myosin primed for action and the bodyguards out of the way, the myosin finally binds to actin, and it is beautiful. beautiful. When they bind, the myosin releases all that stored energy, and in the excitement of it all, the myosin changes shape. It pulls on its precious actin strand, kind of like pulling a rope hand over fist. In the process, it shrinks the whole sarcomere and contracts the muscle. That's the sliding part of the sliding filament model. Now, with its energy spent, that little head has no use for the ADP and the phosphate, so they unbind with it because, remember rule number 2, changing shape encourages proteins to bind or unbind with stuff. That unbinding causes a small change in its shape, which lets a fresh ATP bind there in its place. That binding causes another shape change, but this time it causes the myosin to release from the actin in a tear-jerking scene like some microscopic recreation of the finale from Titanic. But fear not, this epic is not quite over, because this is when the myosin breaks down its new molecule of ATP into ADP and a phosphate, which moves it into the armed position yet again, getting it ready for its next rendezvous. And meanwhile, those calcium pumps are working hard to restock the calcium in the sarcoplasmic reticulum, so they start grabbing the calcium that's floating around, causing calcium to unbind from the troponine. When it unbinds, the resulting shape change puts the tropomyosin bodyguards back into place. It's a circle. Or potentially a big Hollywood franchise with lots and lots of sequels. It keeps repeating itself many, many times every moment while I sit here and talk, and while you sit there and eat and text and take notes, the whole drama replaying itself over and over. Kinda like you'll have to play this video over and over again to get all the little steps of the sliding filament model straightened out. But hey, some stories get better the more you hear them. If you do watch this one again, you will relearn that your smooth cardiac and skeletal muscles create movement by contracting and releasing in a process called the sliding filament model. You'd also relearn that your skeletal muscles are constructed like a rope made of bundles of protein fibers, and that the smallest strands are your actin and myosin myofilaments. It's their use of calcium and ATP that causes the binding and unbinding that makes sarcomeres contract and relax. Special thanks to our headmaster of learning.
Okay. So a lot going on with those proteins. So actin is this thin strand right here, and it's got all these little like binding sites that myosin, which is the thick filament that has all these heads, want to bind to. And the myosin is going to pull the actin towards its center on both sides. And that's what's going to create the sliding in the sliding filament. Now, we do want to be able to like block these proteins. We do want to make sure that like, you know, we can relax our muscles, right? And so we do have like a strand that sort of covers the myosin, I'm sorry, the actin, and it looks like this. That's your troponin and your tropomyosin. Their job is to like block the binding sites so that like you can have like a muscular relaxation, you know. Without that relaxation, you know, we wouldn't be able to unbind our muscles. We would contract once and then we'd be stuck that way forever. So when we're talking about this term here, we call it the sliding filament theory. Or the sliding filament model. What we're describing is where ATP is broken down, and that releases a bunch of energy, and we use that energy to change the shape of these proteins. We cause, you know, the myosin head to reach up and grab the actin strand. That's where it gets its energy in order to do that. So as we're talking about like motor neurons, right? Motor neurons from the nervous system are responsible for making this happen. So this is a term that refers to the neurons. If you look at a motor neuron, we're gonna look at a motor unit actually, you'll see it is where one neuron is sort of running down, and then look at this individual muscle fiber right here, right? You can see this neuron runs down and it interacts with all of the various muscle fibers, right? These are each individual muscle cells that we're seeing here. I know that these rods kind of look like myofibrils. They're not individual myofibrils, right? These are actually, this is a muscle cell, that's a muscle cell, and that's a muscle cell. So this one neuron is interacting with, you know, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 muscle cells. So that is not a huge motor unit, but it's big compared to the pictures I normally pull up. But that's what a motor unit is, right? It's a motor neuron and all of the fibers that it is interacting with. This neuron's interacting with 1, 2, 3, 4 fibers, right? So you've got big motor units and you've got small motor units. So when we talk about like a muscular contraction, we're describing where a motor neuron or a motor nerve is innervating your actual muscle cells. Okay. That's what's happening during a muscular contraction there. Now, if we take a look at where these two, and there's different layers here, by the way, that I don't need you to necessarily know. There's connective tissue layers that are separating all the various parts of the muscle. And because they're separated, there's a little gap between them, which allows materials like fluid, sodium, potassium, things like that to kind of flow between the different layers. Now, the way that the neuron actually interacts with it, you can see here's the tail end of a neuron here. And there is this little gap, and this is your actual muscle fiber. So this is the muscle cell here, and you can see it's got mitochondria, obviously. And then here you can see there's the neuron, and it's got mitochondria. So it also takes a little bit of energy to push this material, we call this a neurotransmitter, from the neuron into the muscle cell. And when that neurotransmitter binds with the muscle cell, that triggers the action potential to run across that muscle cell. So you can actually see that. This is kind of a gross video. If you're squeamish, turn away. But acetyl choline. Maybe that's in, where's the freaking video? Frog. Okay, this is a little bit gross. We're gonna look at the, like, what happens. This is a dead frog. But they're gonna take liquid acetylcholine, which is the neurotransmitter. So these little like bubbles, these little like pink dots here that are moving over to here. Acetylcholine is what binds to the muscle cell, and that's what triggers this contraction. So what we're gonna see is he's gonna take the frog's heart out here. I know it's kind of sad and gross. I think mounting it. Oh no, never mind. I pulled up the wrong video. God dang it. I used to pull this thing up. There it is. So what they're going to do is he's got liquid acetylcholine here, and he's going to pour it on that frog's heart. And you'll notice it's going to start, see all that liquid right there? It starts contracting. The other muscle fibers, he's got the, that's not being exposed here. But like all the tissue that's being exposed to that neurotransmitter is making sodium and potassium move from one side to another, which is how doctors sometimes can use acetylcholine and other materials to like restart different body tissues if they need it to contract. But that's what that neurotransmitter is. Acetylcholine is the neurotransmitter that actually triggers this action potential to run across the neuron, right? So as acetylcholine degrades, it can be recycled, which is really great. This is actually why we need to actually consume choline in our diet. One of our minerals that we consume, if we look at all like the minerals in, trying to find like a list here. Give me a chart. That's not all of them. Major minerals, first trace minerals. There we go. So here's all the minerals that you need a lot of every day, and here's the ones that you need very little of. But like choline, which is not, okay, that's not the whole list. God dang it. There we go. So choline is one of our major minerals. We do need to make sure that we are consuming lots of choline every day in our diet. You're going to get that pretty much from everything. Luckily, choline can be found in pretty much every animal product. It can be found in like Brussels sprouts. It can be found in like cauliflower. Usually very like pungent foods. In fact, you can sometimes tell somebody who's eaten a little bit too much food that's like really rich in choline. It can sometimes cause like body odor, which is not so great. But we do need to make sure that we get plenty of that in our diet, because it's what allows us to, create this neurotransmitter. And that neurotransmitter is what allows us to do everything. By the way, you guys have heard of neurotransmitters. If you talked about like dopamine or serotonin, melatonin, all of those are neurotransmitters that bind with our brain to make us do different things. Acetylcholine is the one that causes muscular contractions to take place. So once acetylcholine is attached to the muscle cell, what that's going to do is that's going to flush calcium ions, Ca2, into this area right here. And that regulatory protein, the troponin and the tropomyosin, right, they are attracted to calcium. So what happens is if it's in this area here, the regulatory proteins move away. And that is what allows the binding sites to be bound, right? That's what allows the binding sites to grab one another, is we flush calcium. So that's what's kind of funny. When we talk about like a neuron innervating a muscle fiber, what's actually happening in that muscle fiber is that it's flushing calcium into the area. And that calcium is what's moving the proteins out of the way so that the binding can actually happen. So those are all the sort of individual pieces in a muscle contraction, right? Acetylcholine gets flushed onto the muscle cell. The muscle cell then runs electricity across its body. And when that electricity gets released, calcium flushes into that area being, we call that the sarcomere, right? So when there is calcium in the sarcomere, the regulatory proteins move out of the way, and then these proteins over here grab, and these proteins over here grab, and they both pull towards their center, and the entire muscle cell shortens. That is every single contraction you've ever made has happened because of this process, which is crazy, right? I mean, like, we are super zoomed in here. So when we're talking about like developing a client's strength, we're talking about like training our clients. What we're really trying to do is we need to make sure that we are keeping we're not only training their muscular system, we're also training their nervous system to be able to control this effectively. And that's what NASM built its entire foundation on. It's all about building like a foundation of stability first, teaching your nervous system how to control your muscular system. Then we can start getting into like heavy lifting later on. And all of that like function is the result of how your nervous system and your muscular system are shaped. Their anatomy. Form follows function, right? So that is sort of what we're looking at there. So the next thing we got to look at is how when we're creating these contractions, what about like the level of strength that's occurring in these contractions, right? When we talk, there's going to be different types of contractions out there. We're going to see two major categories, right? So we are either going to see contractions that create movement, or we're going to see contractions that stabilize and don't create movement or resist movement, okay? So the two types of like movements that we've got, right? We've got the muscle shortening where it bunches up, and we've got the muscle lengthening where it stretches out, right? So the bunching up action, the shortening of a muscle, is what we refer to as a concentric contraction, okay? Concentric contractions occur when the muscle is shortening. So when the sliding happens, and this is going towards its center like this, right? That is a concentric contraction. This muscle is shortening, right? And so concentric contractions occur when your muscle is generating enough force to overcome some type of resistance. So like if I'm in the bottom of a squat and I contract to come out of that squat, right? That's a concentric contraction, right? I'm shortening my muscle fibers and moving my bones to overcome a resistance. If I bring my arm up towards, my shoulder here, that's a concentric contraction in my bicep. But there's exactly the opposite happening on the other side. If we flip to the other side of the joint, the tricep muscle is relaxing. That's an eccentric contraction. Eccentric contractions occur when the muscle is lengthening. So like if we were tug of warring, or I'm tug of warring, this is not tug of war. If we were arm wrestling, If I were arm wrestling with somebody in class right now, right, and they're stronger than me, and they're overcoming me, I'm trying to create as much, create as much force as possible. But whoever I'm arm wrestling is creating more force than I can, right? So they're concentrically shortening while I'm trying to concentrically shorten, but unfortunately I'm not strong enough. So I'm eccentrically lengthening, right? And that's what happens in like a tug of war, or God, in arm wrestling, right? If you are concentrically pulling on one side of the joint, the opposite side of the joint will naturally eccentrically lengthen. Now, this term is probably one of the most, or this concept is probably one of the most important concepts that we talk about as trainers, because when we're looking at human movement, let's see, we see like, let's see if we can find like a video of somebody who, squats improperly. No, that's not it. There we go. Let's say we see somebody who squats like this, right? This is they're trying their hardest, to squat properly, but unfortunately their knees kind of look like this. Their knees buckle inwards. What that means is they have excessive concentric shortening on one side of the joint, and they have limited concentric shortening on the other side of the joint. What's happening here? Oh, that's not the part of it that I want. Okay, I'll just draw one. The knees are caving in kind of like this, right? In this angle here. Imagine this is, that lady's knees, right? So what's happening is they're both caving inward. So we've got excessive concentric shortening this way, and we have limited concentric shortening that way, right? This muscle that's on this side of the joint that's supposed to pull the knees outwards is being eccentrically lengthened against its will. That's why we call this muscle that's concentrically shortening, we call that an overactive muscle. It's too strong. And then we have an underactive muscle on the opposite side of the joint. It's not strong enough. It's too weak, right? So what we're trying to do to fix somebody's posture is we just look at that anatomy and we go, okay, what's the weak muscle? What's the muscle that's being pulled, against its will? In this case, it would be the gluteus medius here on the outside of the butt there, right? So we would want to strengthen that muscle, and we want to loosen up. This is going to be the adductors that are pulling this way, right? And that's what's happening in somebody whose knees are caving in. We look at knee valgus. What's happening is somebody's knees are caving. That's such a bad picture. What we're seeing is like their knees are caving in because these inner thigh muscles are squeezing too concentrically hard, and the glute muscles are not squeezing concentrically hard enough. They're being eccentrically lengthened, right? They're losing the arm wrestling match, right? So that's the two movement types. We've got concentric, which is shortening. We got eccentric, which is lengthening. And then we've got isometric, which is going to be when the muscle is remaining at a constant length. So one of the other reasons that could cause somebody to causing somebody to maybe like knee wobble a little bit while they're squatting. I'm finding a GIF of this maybe. Wow, that's actually, okay, so this is actually great. She should be doing this. But let's say you see somebody who's like, I'll just do it. We see somebody who's squatting, right? And they kind of squat like this. They just have like, they're not like really like fully caving in, right? Sometimes it's just a stability problem. You see like people wobble a lot, or think about like when you're crunching, sometimes you get that like quivering sensation, right? What that is that's your body trying to figure out exactly how much force to generate to get you to stay in place. It's trying to figure out how to make a stabilizing isometric contraction. If I'm trying to stay put, that's an isometric contraction. So a plank, a wall sit, all of that is isometric. It occurs when the muscle is developing an equal amount of force to the resistance, right? So this jar weighs, I don't know, we'll say 1 or 2 pounds, right? Probably 1 pound. Probably less than that actually. But so if I generate 1 pound of force, I'm going to overcome this weight here, right? And if I create less than 1 pound of force, the weight's going to overcome me. So I'm going to concentrically shorten if I generate more force than the resistance I'm working out. I'm going to eccentrically lengthen if I generate less force, or I could hold it in one spot. And that's an isometric contraction. So if I create exactly the right amount of force to stay put, that's isometric. So when we're talking about like training our clients, we need to look at like how we can get them to create a strong enough isometric contraction to stabilize their body while they are moving other joints through specific ranges of motion, right? Like if I can't stabilize my core while I'm trying to do like a shoulder press, right? What'll end up happening is I'll probably end up arching my back, right? Or I might do this, or I might twist to the side. You'll see people, you see it all the time. It's like their shoulder, they're struggling to get the weight here. So then they end up, their body's like, well, let's try this instead. It tries to leverage the strength necessary from other places. And that's going to cause some type of overuse injury. I mean, look at like, look at what happens when somebody is like squatting and they do like a little bit of like a butt wink, right? There we go. So holy crap. So this girl's already got kind of an anteriorly tilted pelvis. And then she's squatting down, and see how like her pelvis is rotating downwards? It's actually rotating back into a normal position. So she's got an excessive low back arch. You can kind of see that she's got way too much lumbar arching happening there, right? And then as she gets to the bottom, you might think that she's got like a posterior tilt, but it's not posterior. It's actually just getting back to neutral by the time she gets to the very bottom there. So why is her pelvis anteriorly rotating, right? Well, you think about your pelvis, it's kind of like a steering wheel. And so what's happening is it's being pulled down this way and being pulled up this way, right? So her hip flexors are pulling down and her low back is pulling up. Both of those muscles are tighter than they need to be. So what do we need to do to fix it? We're just going to flip these arrows around. We need to strengthen this way and this way. That's going to be her glutes and her abdominals. If she strengthens her glutes and her abdominals, that arching is going to go away. That's how we're going to analyze posture. And all this comes from our understanding of concentric contractions. So, it could be that, maybe she's got a bad nervous system connection. She doesn't know how to use her core properly. So what we need to do is we need to kind of come up with an exercise that's going to teach her how to wake up her core, right? And teach her how to wake up the glutes, right? And we need to teach the low back and the hip flexors to shut down. We're like, hey, you're on too often. Let's turn it off, right? How do we do that? We're going to get into that more in the second half of this course, but we can strengthen by doing exercises specifically targeting the abdominals and the glutes by themselves. That's going to wake up the nervous system and teach the nervous system how to control those things. And then we can stretch muscles, which is actually a good way to shut those muscles down and turn them off. All right, we're almost wrapped up today. Any questions so far? Al, Silos, how you guys feeling?
Okay, quick question on the posture on the squats that we just saw right now. Mm-hmm. I have, one of my buddies, I think he strained his back on a squat that he was doing. Mm-hmm. And he was asking me, because I told him that I'm going to school, what I'm doing right now. Yeah. So I told him, I don't know nothing yet right now, but I'm learning. It's just like beginning of the process. I go, but out of what I used to do, before I started doing this school stuff, I told him, how were you posturing when you went down to squat? So he said he was slouching. Like he was like, he had the bar, he had the bar, the bench bar on his back. Just kind of and that instead of being up straight, he kind of had like a little slouch to it. And when he pushed up, he strained, I think the lower part of the back and the middle part of his back.
So if you collapse in the top of your squat, so like if you collapse a little bit here.
Yeah, that's exactly what he did right there.
Yeah, what that'll do is that naturally flattens this out and it takes away his lumbar curve. So what he needs to do is he needs to actually pull down on the bar while he's squatting. If he gets a little bit more active contraction there, that will pull his shoulder blades and keep his chest up. Then he just needs to make sure that he maintains a neutral pelvis. He doesn't need to arch back. That's what a lot of people will try to do.
Yeah.
They'll try to like go like this. That creates exactly what we just got done seeing in that lady. Too much of a low back arch. That's also not good. We want neutral. We want a neutral spine. So tell him to rest. Get the pain to go away.
yeah, that's what I told him. I was like, rest up, man.
Yeah, and then he needs to work on his core strength. That'll help him stay upright and more naturally. And then be more active by pulling that bar into it. If you pull down on it, it'll actually create activity, which will keep him upright.
Oh, okay, cool.
Yeah, that's a classic.
Thank you.
People goof that up all the time. I had a really bad habit of that when I was younger. Any other questions, guys? Anything else? Oh, Al, I can see you're unmuted. Do you have a question?
No, I'm good.
Oh, okay. I was like, he's unmuted. I couldn't hear him. Okay, cool. All right. So, guys, let's zoom out. let's zoom out just a little bit, right? Let's stop talking about like microscopic proteins. So let's take a look at a muscle in general, right? You're gonna see like different muscles. I mean, look at just like a steak, you know, different types of steak, marbling. You know, if you look at like the various types of steaks that are out there, you can see like there's like a big fatty section and, you know, lots of marbling there. And then there's other types that are like, you know, just look at this one. This has like no marbling at all. Like that's like just pure protein. You know what I mean? So that is going to be the difference. You can see different levels of marbling there, right? That's going to be the difference between like, you know, with no marbling at all. There's no fat in that. It's pure muscle. That's probably a very, 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 very strong fiber. This is probably coming from the cow's strongest muscles, right? So that's why different cuts of meat have like different fats. So the meats that we generally tend to like the most are the ones that are like really fatty cuts, you know, at least, you know, most people say that's where all the flavor is, right? So that is true of your muscles as well. There's going to be very different characteristics found in your muscles. They're going to look visually different because they have different equipment. Again, form following function, right? They're going to have a different shape, which is going to allow them to do different things. So we're going to break muscles into two major categories. You are going to have your type 1 fibers. We talked a little bit about this yesterday. These are known as your slow twitch fibers. More specifically, they're called your slow twitch oxidative fibers. What that means is like, think slow twitch, think weak contraction. And think oxidative, think super long, able to spit out energy super consistently contractions. So your slow twitch oxidative fibers are muscle fibers that have a very well-developed respiratory system. They are packed full of mitochondria. And that ensures that they can deliver oxygen to the muscle fiber very easily, and they can just spit out energy constantly, right? So they are smaller in size, you know, type 1 versus type 2 muscle fibers. You're going to see the difference between like, you know, a sprinter versus a marathoner in terms of like how their body works. That's
I think you turned on your mic.
What's that? Oh, Eric, what'd you say?
No, my bad. Sorry, my mic turned on.
Okay. Sorry. So when you look at like the type, look at these kind of this kind of picture here. You can see like this type 1 fiber is like bright red. Okay, that's actually what you'll sometimes hear these referred to as. You'll hear them referred to as your red fibers. And they call them that because they get a lot of blood flow, and blood means oxygen delivery. They have a lot of things that can use that oxygen, like mitochondria. There's actually going to be more enzymes there. So you can see there's actually more oxygen within the mitochondria, whereas over here, there's actually less oxygen, there's less mitochondria, there's less enzymes, right? So your type 1 fibers, you want to think of as like your endurance fibers. if you look at like the difference between like a runner and like a bodybuilder, right? Runners have these big, long, lean muscles. So they tend to be smaller in size, but runners have a lot of endurance, right? So they have more mitochondria. They're also going to have greater hemoglobin levels, right? Their red blood cells probably have a lot of hemoglobin in them, so they can grab oxygen, giving them a very high VO2 max. They got greater myoglobin levels, meaning that they're able to carry oxygen to the muscles themselves. And these are the types of muscles that we use for our long-term contractions. So everybody in here, we all have a mixture of type 1 muscle fibers and type 2 muscle fibers. Your type 1 muscle fibers are the ones that are keeping you upright right now. You've been using these, you know, my jaw is very type 1. You know, I've been talking all morning, right? Your core muscles that are keeping you sitting upright, those are very much type 1. Your heart, God, that's like type 1/2, you know, like that's really type 1, you know, packed full of mitochondria, packed full of enzymes, packed full of energy producing, right? So those are your type 1 fibers. Type 2 fibers, these are the ones that you're probably thinking of when you think of like sports. When you think of like an athlete, right? You're probably, and like when you think of like you being very explosive or lifting weights in the gym, that's where we're talking about our type 2 fibers. So type 2 fibers are going to be known as your fast twitch fibers, okay? So fast twitch fibers are muscle fibers that have a lot more calcium in them. Remember, we need to flush calcium into the area in order to create like a big contraction. And they have more cellular volume. There's actually going to be a lot more fluid in these muscle cells, right? If we look at type 2 muscle cellular volume, you'll see there's like a lot of empty fluid space in here, right? So here's kind of the difference between like a type 2 muscle fiber. And, oh, that's actually showing, oh, that's kind of what I was looking for, but that actually is a picture of a diabetic. That's showing like how glucose can get stuck in the cell and suffocate it. Well, you know what I'll do? We'll just kind of go to our picture here. Muscle. So, there we go. That's a sperm cell. God, why can't I find the pictures I want today? That'll work. There we go. So this is a picture of cardiac muscle, which has tons of mitochondria. Look at how like this, there's just mitochondria straddled all over the place in this particular cell. All that mitochondria makes this very good at spitting out energy. That's a type 1 muscle fiber. A type 2 muscle fiber would have a lot more space and a lot more cellular volume. So the fibers are going to be much bigger. You'll actually see type 2 muscle fibers. And that's sort of the difference. I mean, you look at a runner versus a bodybuilder, you know, you look at their body sizes, they're very different looking, right? Like this is these very long, you know, lean muscles here, right? Versus like these very large, very bulky muscles here. So what you're going to see, the difference between these two, right, is that, you know, one of them is producing energy in the mitochondria. That's our aerobic metabolism, very good for long sustained energy production. And then the other one is very good at cellular volume energy production. So we're producing energy in the fluid of our cells, which you guys will remember is glycolysis. Glycolysis is where we produce energy in the fluid of our cells, right? Sugar goes in and then it becomes energy, right? That glycolysis is how we create lots of big bursts of energy, but we can only keep it up for so long. Unfortunately, we get exhausted from that very, very, very quickly. Yeah, question.
Just want to know, like, I don't know if it's going to this topic or not, but just like, because you show us the bodybuilder, just a quick question. Is it the truth that most bodybuilders don't know how to swim, or they don't do that because they just like cannot hold themselves because they're too heavy? They cannot hold themselves on the top of the water and they go up in the bottom. Sorry to say that, but I heard yesterday exactly that was one of the conversations I had with my friend, and that's what I'm asking. Hello?
No, I mean, I don't know. Bodybuilding, you won't catch very many bodybuilders swimming simply because it's cardio, and most bodybuilders avoid cardio because it burns so many calories. And part of bodybuilding is ensuring that you have like a positive amount of calories instead of like a negative amount of calories. So they're trying not to burn calories most of the time as a bodybuilder. So that's probably the biggest reason why you won't see. It would not make it any harder to swim. I mean, yeah, their body would be heavier. But I will say this, the more body fat you have, the easier it is to swim because you're going to be more buoyant. So fat underwater weighs a lot less than muscle does underwater, right? So that's actually one of the ways that like hydrostatic weighing works to test your client's body fat percentage. We know how much like a pound of muscle weighs on land. It weighs a pound, and we know how much it weighs underwater. We also know how much a pound of fat weighs on land, and we know how much it weighs on water. So if you weighed 100 pounds or 200 pounds on land, and I don't know what the numbers are, but we'll make this up. We'll say you weigh 50 pounds underwater, and that tells us that you have 20% body fat. And then you take the same 200 pound, or take a different 200 pound person, they weigh 200 pounds on land, and they weigh, let's say, 25 pounds underwater, right? So they weigh less underwater than the first person did. That would tell us that that person probably has like 30% body fat or 40% body fat, right? So like that fat is very buoyant. So like a lean bodybuilder would have a hard time staying on top of the water, I guess. But like it wouldn't have anything, I mean, like it wouldn't be any different than like an Olympic swimmer. I mean, you look at Michael Phelps, right? Like Michael Phelps is very long, very lean. He's an incredible swimmer, right? And he's very, very muscular. So could he be more buoyant if he had higher body fat on him? Yeah, but then he would also have like a higher body weight, which would give more drag under the water, which, you know, he would float easier, but he would have a harder time moving forward.
Moving fast,
right? Because there'd be more drag. And then he would also struggle endurance wise, you know, well, he wouldn't struggle endurance wise. He would struggle like power output wise, because fat is used for long duration exercise, whereas swimming is usually like a short duration, like sprint at an athlete level, at least.
Okay. Thank you.
Yeah. So I wouldn't, if anybody said, I wouldn't pay any, like if anybody told you that bodybuilders can't swim because they're too muscular, I wouldn't put any stock in that.
Because I was laughing at them. Like, that's why I didn't like
that doesn't make any sense to me. I will say, I do have a fun story though about a bodybuilder who swims. I used to train this guy who was a cinematographer, and he shot Pumping Iron. So I trained the guy who shot that movie, the Arnold Schwarzenegger documentary. And he worked with Arnold Schwarzenegger twice.
No way.
Yeah, so he
That's
worked
one of
with
my favorite movies, Pumping Iron, all of them.
It's great, right? Yeah. So my client Victor, who was in the Navy, he was like a World War II Navy veteran, right? I trained this guy, by the way, when he was 84. That's when I started training him. Like, he got a trainer in his 80s. He was in pretty good shape before that, but like that was when he was like, I'm getting aches and pains finally. And I used to train him and his wife. And so Vic, you know, after he got out of the military, he got out of the Navy, he became like a cinematographer. He shot Pumping Iron. Years later, he worked with Arnold again and shot Jingle All the Way. Yeah. And he said that he remembered him, and he was the nicest, and like Arnold is like one of the coolest, awesomest dudes to be around. But
I used to see him all the time in Ocean Boulevard.
Yeah, absolutely, down in Venice. So here's the thing though, in his younger days, he was extremely arrogant.
Yep.
Of course, how could he not be? You know, like, how could he not be? So he and his buddies were sitting around. They were on the set of Pumping Iron, and they were on a beach. And he goes over to Victor and he's like, "You see that island over there? How fast do you think I could swim to that island?" And Victor's like, I don't know, Arnold. Like, I don't know. And he's like, "I'll bet I'd be the fastest swimmer ever." Like, he's like, "Look at how muscular I am." Like, and Vic's like, I don't know. He was like, "I'll bet you $100," which, you know, this is back in like what, the early 70s.
That was a lot of money.
A good amount of money, right? He's like, "I'll bet you $100 that I can swim to that island and back before you can." And Vic's like, "Oh, Arnold, like, I was in the Navy." But Vic's like he's a small, like, you know, skinny dude. And he's like, "But I was in the Navy, Arnold." Like, so I'm a pretty good swimmer. And he's like, "I'll bet you $100." He's like, "All right." And so Vic just demolished him. Like, you know, sprints of it. Arnold gets to the island, and they were supposed to touch the island, turn around, and swim back. And he like flops down on the sand, and he's like completely gassed, totally exhausted. And all of his other buddies, they had to take a rowboat over there and like pick him up. And he was really embarrassed. But it's like, so that, but you know, as that bodybuilder, you know, maybe not so.
That's
great
why you don't go. That's why you don't go up against nobody who was in the Navy.
Yeah, right. Yeah,
I even know better.
right. So that is like a fun. Yeah, my client told me that story years ago, and I remember that as that is a good example of a bad swimming bodybuilder.
Okay.
I wouldn't call that a rule. No. So.
Okay, thank you.
Yeah, no problem. So those are our type 2 fibers. So the type 2 fibers are going to be, you know, larger in size. And then they're going to have lots of proteins in them. So what I want to do is I want to go show you just a picture of the difference here, right? So we'll go ahead and put a little line here to kind of separate. So here is, we're going to kind of separate this out a couple different times. We're going to draw 3 different muscle fibers. So here's a normal muscle fiber in an untrained person, right? So this is our untrained individual, somebody who, you know, hasn't necessarily exercised, right? So what we're going to see is we're going to see some proteins in there, right? They're going to have myofibrils, you know, those are their myofibrils right there, those little rods, right? But they're also going to have like little mitochondria, you know, maybe they'll have like a couple little mitochondria in there as well. So there's a little mitochondria, there's a little mitochondria. They'll have all the other normal stuff too. So all this empty space is cellular fluid. Okay, so then let's look at a type 1 muscle fiber. So a type 1 muscle fiber is going to look just like this, except that it's going to have a whole bunch more mitochondria. We're going to pack this thing up with mitochondria. So this muscle cell is really good at spitting out energy, right? This one's just spitting out energy left and right. Okay, so there's our, and I'll actually, let's go ahead and fill this in just to make this. So there's our myofibrils, right? And then there's our mitochondria. We'll go ahead, oops, make these this color here. Okay. All right, so there we go. Now we got our myofibrils and we got our mitochondria, right? So that's a type 1 muscle fiber. We have more mitochondria in there. Now, in a type 2 muscle fiber, we're going to see 2 different versions. We're going to see type 2A, and we're going to see type 2B or 2X. I'm not sure which version you guys are going to have listed. The world changed that. Some of them, sometimes it's type 2A, sometimes it's type 2B, sometimes it's type 2A and type 2X. X for extreme. I don't know which is the right term anymore. They keep changing it. They're the same thing though. So let's take a look at this type 2A muscle, right? So what we're going to see here is we're actually going to notice that like this is a much larger muscle cell, okay? So this is a much larger muscle cell in type 2A. But the reason it's larger is because it still has the same organelles in it, but look at how much more cellular fluid there is, right? Like there's just more empty space, right? And that empty space is where the energy production is happening in a type 2A muscle. So we call type 2A muscle fast twitch because it is fast twitch, but it's also oxidative glycolytic fibers, meaning that like in all that fluid, it's able to output energy relatively quickly and relatively good. You know, we might even see a little bit of extra mitochondria in here, you know, we might see just like a little bit more, right? But we're not going to see a ton. We're not going to see as much mitochondria as a type 1 fiber. But we're going to see more cellular fluid. So this is pretty good at energy production, but not as good as energy production as like a type 1 muscle fiber, okay? So that's a type 2A fiber. Type 2X fibers are going to be the same size, so they're going to be really big, but they're going to have more proteins inside of them. So those are our myofibrils, right? So the reason that these muscle cells are bigger is because there's more proteins, there's more rods in the muscle cell. And then, yeah, there's a goodly amount of, there's probably a little bit of mitochondria in there as well. We're definitely going to see some mitochondria here and there, but not nearly as much as we would see in like a type 1 fiber. We also don't see as much cellular fluid in a type 2X fiber because all the fluid space is being taken up by the proteins. So this is a true fast twitch explosive fiber, okay? Whereas this is like a fast twitch semi-endurance fiber. So like this is fast twitch, but it also has like a little bit of endurance. And this is a slow twitch high endurance fiber, okay? And that's what that's what slow twitch means. So like if you think about the difference here, right? Over here on the right hand side, this is like an Olympic weightlifter or a power lifter, right? And this over here is your classic bodybuilder, okay? And this person here is like your runner. And this is somebody who hasn't trained, right? So that's the difference between type 2A and type 2X fibers. So your type 2A fibers your type 2 fibers in general are all fast twitch. Type 2A is fast with a little bit of endurance. Type 2X is fast with very little endurance. Typically, when you're training athletes, you are probably focused primarily, and when I say athletes, I mean like field sport, court-based sports like basketball, football, baseball. You're probably training for much more type 2A muscle fibers. That seems a little weird. You might be like, well, wait a minute, don't we want them to be as explosive as possible? Wouldn't we want type 2X? Yeah, you would. You do want them to be explosive. Like if I'm training a baseball player, right, their oblique muscles, I want full type 2X. I want them to be as explosive on like a twist as they're swinging a bat as possible. I want a pitcher to be as fast, explosive twitch as possible. But I also need them to have some endurance because they got to run the bases. They got to be able to be in the outfield, right? Football players, a running back. Yeah, I want them to be really explosive, but I also need them to do more than one play per game. So generally power lifters, you'll see, football players be able to put large amounts of weight on the bar while they're lifting, but they will not be able to put anything in the weight room compared to like an actual power lifter, like a strong man, you know, like Eddie Hall, Thor Bjornsson, you know, Brian Shaw, all those like full power lifters, strong men. They are true fast twitch athletes, type 2X. So when we have an athlete, we probably want a pretty good blend of those things. And that's where you're going to train different ones, right? You've got like full powerlifting, like maybe you've got like a snatch all the way from the ground up over your head, you know, or a clean and jerk, right? Very explosive, very good for type 2X fibers. But you want to keep the reps below 5. You want to keep those reps really low, because then you are training type 2X fibers. If you start to get into 12 reps, you're getting into like type 2A category. You start getting above 12 reps, you start getting into type 1 category. All of it is good. All of it is honestly good. We love this. And for the record, NASM believes that it's very safer to build a high percentage of type 1 fibers at first, so you get a lot of endurance, you build a foundation of endurance, then we go up with the intensity as we climb up the OPT model, right? As we start getting into strength instead of endurance, we're starting to focus on type 2 muscle fibers. We start with type 2A, and then eventually when we get a peak athlete, we start going for type 2X. So you think about like growing a muscle, you might start by building a little bit of your endurance, and then you start focusing on building up the size of your muscle cells. You start limiting your, you start giving yourself more rest, start lifting a little bit heavier, start producing more lactic acid, right? Because lactic acid gets produced in this fluid, right? That's the byproduct of glycolysis. Then eventually we start, increasing our strength, and we just start, making more of those proteins. We start shoving more proteins into the cell. The more proteins you got, the stronger your fibers are. So that's why NASM starts our clients. We always encourage us to start our clients off with high repetitions and then move to low repetitions. We start with low intensity and we move to high intensity. Whew. So a lot of anatomy today, a lot of terminology today, but hopefully you got a pretty good understanding of, how our muscles are working on a zoomed in level. Any final questions before I let you guys out of here?
No, that's pretty good.
Quick one. How long?
How long?
Oh, my bad. Go ahead.
No, please go ahead.
Oh, how long would you recommend on stretching before heavy lifting?
No, that's fine. Go ahead.
Oh, before heavy lifting. Yeah. So before heavy lifting, you want to only stretch for maybe 1 to 2 seconds, and anywhere from like 5 to 10 repetitions. So if you're stretching out your chest, you would basically like pull it back 1, 2, and then relax, and then do it again, and then relax. So you're going to do it for reps rather than doing it for like a long duration. You'll accrue, if you do 2 seconds each and you do 10 reps, you'll accrue like 20 seconds of stretch. You know what I mean? But don't hold it for 20 seconds, because if you hold it for 20 seconds, you're actually kind of inhibiting the neurons. That has been shown to decrease neuromuscular activity. So it's a good way to get, you want to stretch before you work out. Like people say, don't stretch before you lift. That's bad advice. We like stretching. We just want to make sure we do the right type of stretches. So those are called, you can look this up. Those are called active isolated stretches. I'm going to teach you guys that at the end of the mod. Actually, we're going to teach that probably Thursday next week, actually, because it does come up in this class as well. But there's different types of stretches out there. There's static stretches, which is what you usually think of, the big long ones. And then there's active stretches. And then even just like arm swings, right? We call that a dynamic stretch. That's
what I do before I power lift heavy. I do that. I do like the swinging of the arm like that, and then I do the one you were showing right now like that. Yeah. And my buddy always tells me, "Why you stretch? Just hit that weight."
Yeah. No. Always stretch. Get the blood.
No.
Yeah, you gotta wake. It not only does it wake the muscle up to keep it safe, it also like gets the energy production going, which will make your lifts more effective.
Hell yeah. Cool. Thank you.
Yeah, of course. Yeah, that's a good one.
So Brad, is it for any lifting or just the heavy lifting? Like before you lifting the weight, you got to do the stretch, the one that you just said, or just for heavy lifting,
So
like
heavy lifting,
weight lifting?
Yeah, for heavy lifting, we do the active stretches. For light endurance type lifting, like if you're going to go for a run, go for the static stretches, the big long duration. If the intensity is really high, the stretch duration should be low. If the intensity is really low, the stretch duration can be really high.
Okay.
So,
all right.
Yeah.
Thank you.
Of course.